Hello everyone, and thank you for being here today, and hope you're all staying safe. My name is Daniel Schloesser. I'm a fifth-year PhD student in the Cognitive and Information Sciences program at the University of California, Merced. I'd like to start off by saying special thanks to Derek Hollenbeck in the Mechanical Engineering Department and Christopher Callow, my advisor in the CIS department, for all of their time and effort throughout this project. I'll be speaking to you today about social foraging in groups of search agents with human intervention. Foraging is a problem that every species needs to solve. Now, how they solve this problem varies widely and can change based on context. But importantly, foraging also serves as a convenient example of a task that can be completed independently or collectively. For instance, here we see a gannet flying by itself engaging in an independent behavior. Alternatively, we can see the same species clearly engaging in a collective strategy. Now, gannets are just one example, but there are obviously many species that can vary their foraging strategies. And we are a great example of a species with complex behaviors that permit switching between individual and collective strategies. We do it all the time. We can easily go from searching in a group to searching independently. This matters especially in a situation like foraging because the strategy that gets used can directly impact the survival of the searcher. So it's important for us to know more about how the connection between searchers can change that outcome. Now, humans, among other complex species, engage in something called loose coupling, which allows them to flexibly switch between independent and collective strategies. And we're interested in how cooperative outcomes like performance can change because of the bond between cooperative searchers. So striking a balance between independent and collective strategies could potentially improve the performance of the whole group. To better understand the impact of role switching, Harrell et al. 2017 serves as a great reference. Here, the researchers investigated the foraging behaviors of Israeli griffon vultures. They were interested in seeing how social cues influenced group foraging behaviors among the vultures. To do this, they tracked the movements of tagged vultures and recorded information collected at the shared roosts. The researchers were then able to break the vultures up into two groups, informed and uninformed. An informed vulture was one who showed visual signs of recently having found a food source, and the uninformed vultures were those who did not. Panel A shows the flight paths of two vultures, one informed and one uninformed. What we can see is that the informed vulture's path is closely followed by an uninformed vulture. Alternatively, in panel B, we have two uninformed vultures, both seemingly reaching the same target but not together. What Harrell et al. 2017 found was that the informed vultures were far more likely to be followed after leaving the roost than those who were uninformed. This tells us that the foraging strategies of griffon vultures changes dependent on social cues and that they can flexibly switch between independent and collective foraging strategies. So why is it good to switch roles? An answer to that is switching roles allows a group to flexibly change their strategy depending on the situation. Take honeybees studied in Sealy 83, for example. This study recorded foraging behaviors of the bees and how that changed depending on food scarcity. Here the behaviors of the bees were recorded across two time periods, one when available resources were low and the other when they were high. What we see is that when food resources were low, scouting behaviors were high. Alternatively, when food was plentiful, recruiting other bees occurred more frequently. This study showcases how the availability of food resources can drive which cooperative strategy gets used, where the needs of the colony direct the foraging strategies that were used at different times of year. Our aim was to create a system that could simulate more independent or collective foraging strategies. Here we wanted to create a scenario involving searching agents whose behaviors could be systematically manipulated. So we created a loosely coupled system where the individual components were not dependent on one another and could function in the absence of the other components. But when these components were active at the same time, interesting behaviors could emerge out of that system. For our model, when all three components of the model are present, we create a collective, loosely coupled system of foraging agents. Now, our agent-based model consisted of 10 agents searching at a time, whose goal is to find as many gold star targets as possible. There was only ever one target present at a time, and as you may have noticed, once an agent finds the target, it takes time to consume it. Now, importantly, the more agents that find the target, the faster it can be consumed, so there is some advantage to finding the target alongside other agents. Once the target was fully consumed, a new target would randomly generate at a new location, and this would occur throughout the entire trial. The first component that we manipulated was a flocking parameter. This flocking parameter averaged the agent movements to determine their next movement. So for the agents that they could see, this parameter averaged the movements of those other agents, causing the entire group to flock. The second component that we created was a distancing parameter based on the Lennard-Jones potential. This potential creates a push and pull function connecting the agents together. The agents don't want to be too close nor too far from one another, so this component primarily cares about how far away the other agents are from those that they have in view. And the last component of our agent-based model is a correlated random walk. As you probably noticed in the previous slide, the agents in the distancing condition were moving quite randomly. That's because they mostly were. This random component serves as a sort of baseline and is present in each of the other three movement conditions. Taking a bird's eye view of the agent movements, we can see that agents' movements sometimes changed considerably depending on the active components. We can also see some noticeable similarities. It's important to note that the loose coupling condition seemingly covers the space most uniformly, while conditions like the flocking condition look super diffusive. Here is an overview of how each component worked. Now, there are only two more components left to talk about. The first is the chaining parameter. This parameter directed agents who have detected the target to turn red and head directly towards the target location. Now, the chaining part refers to those who have seen an agent find the target, but don't see the target directly. If an agent saw another turn red, they would turn towards that agent and follow them towards the target location. This caused multiple agents to link together in a chain, but only locally. This link could not cascade to other agents down the line. And lastly, we have the player's experience. The player controlled their agent by simply using the mouse. When moving the mouse cursor, their agent moved at a constant speed towards the mouse cursor's current position. Importantly, the player's agent is not influenced by the movement components, but the other agents interact with the human like they would any other agent. You can see a green circle around the player's agent. This is their field of view. Everything outside of that circle is concealed. So in order to find targets, the player would have to actively search the space alongside the other agents. Our data collection consisted of two independent parts, one where all 10 agents were simulated, and one where 9 agents were simulated alongside one human player. In total, 60 UC Merced students completed each of the 4 movement conditions. We also ran 60 simulated batch runs of only simulated agents for a comparable sample size. Now, the first thing we wanted to know was how the movement conditions influenced measures of global performance. On the y-axis, we have a combined measure of the average search time and consumption time for each condition on the x-axis. What we see across the conditions is that the loose coupling condition has the lowest search time and consumption time overall, resulting in the loose coupling condition having the best overall distribution of times. This is also supported by the loose coupling condition finding the most targets overall. So with only simulated agents, the loose coupling condition already appears to outperform the other conditions. When we look at the distribution of search and consumption times with a human involved, we see the same pattern. Overall, the loose coupling condition performs the best. This again is supported by the amount of targets found in each condition, but with the aid of a human player, each condition uniformly finds more than what the simulated agents alone were capable of finding. But regardless of whether or not the human is present, the loose coupling condition outperforms the other movement conditions. Now that we have some information about global performance, next we wanted to understand how the agents were grouping together. Here we have a probability distribution for each movement condition for both datasets. Each distribution corresponds to the probability of having a certain number of agents in view. We don't overtly see too many differences across the datasets, but we do see significant differences across movement types. What these distributions initially tell us is that the loose coupling condition provided the most variable range of possible grouping sizes, while the flocking condition is very static. Now, to know more about the grouping variability of the searching agents, we conducted an entropy analysis on the variability of the number of agents in view during the searching period of the task. What we can clearly see is that the flocking condition displays almost no variability. This finding shouldn't be surprising because the agents in this condition were always grouped up, regardless of whether the human was present or not. What is important here is that the increased variability in the loose coupling condition is a likely reason for the increased global performance. Agents were more likely to be searching for targets across several groups, and when they found the target, they were able to consume it cooperatively, which allowed them to consume it quicker and able to move on to the next target sooner overall than in other conditions, something that other conditions like the flocking condition were unable to do. Lastly, now that we understand a little more about how agents are grouping up in each condition, we wanted to know more about how they are covering new space while searching for targets. To do this, we created a measure that takes the amount of new space that was covered between consecutive targets and divided that number by the amount of time it took them to find that target. This produced a rate measurement we call area search rate. This measure provides an indication for how well the agents are covering the space and how long it takes them to do that. If the agents are able to cover a lot of space in a short amount of time, then the area search rate should be high. If it takes a long time to find a target, or not a lot of space gets covered on average, then this rate measure should be low. One last thing, for this analysis, we conducted it on only one simulated agent across both datasets. We did this because we wanted a measure for how an individual was searching the space and whether the human could impact that area search rate. What we see is that, in fact, the human does impact the search rates of the other agents across all non-random movement conditions. This is a really surprising result because the agents in both datasets followed the exact same movement rules. But despite this, the human was indirectly able to influence the searching rates of the other agents when those other agents could be sensitive to the human's movements. Simply put, the agents improved by working alongside a loosely coupled human. Now, up to this point, we have been looking at measures of the agent's performance, but how well did the human cover the space? Unsurprisingly, the human consistently searched the space at an increased rate. This makes sense because the human moved exactly the same in each movement condition. What is interesting is that the human search rate improved when working alongside the loosely coupled agents. Because the agents in the loose coupling condition moved in more predictable and interactable ways, the human was truly able to work alongside them, which likely helped them to search the space most effectively in the loose coupling condition. To wrap up about the loosely coupled agents, loose coupling increased collective foraging performance across all measures of global performance. This was likely the result of that condition being able to appropriately balance searching and consumption times. Also, loose coupling diversified the groupings of those agents. Agents were actively searching for the targets in more possible group sizes than in the other conditions, and this increased grouping diversity likely helped to improve their overall performance. And conclusions that we can say about involving a human include that they improved the searching effectiveness of the non-random agents, which surprisingly allowed the agents to search the space more effectively. Along with this, the human also searched the space most effectively when cooperating with loosely coupled agents, increasing their own area search rate, likely benefiting from the stability provided by loose coupling. And with that, I'd like to thank you all for your attention, and I'm happy to take your questions. Thank you.